Synthetic Antibodies, narrated by Luke McLaughlin, biotech digital marketer, business developer, and life science content creator. Synthetic antibodies represent a burgeoning field in biotechnology, characterized by the design and production of antibody mimics that can be generated entirely in vitro. This eliminates the need for animal-derived antibodies, thus paving the way for more ethical, sustainable, and possibly more effective therapeutic and diagnostic tools. Synthetic antibodies encompass a broad spectrum of technologies, including recombinant antibodies, nucleic acid aptamers, and non-immunoglobulin protein scaffolds, which can be engineered for high affinity and specificity to virtually any desired antigen. Recombinant antibodies. Recombinant antibodies are a cornerstone of biotechnology and therapeutic medicine, offering a precise and versatile means of targeting specific antigens for both diagnostic and therapeutic applications. Unlike conventional antibodies obtained from animal sources, recombinant antibodies are produced using genetic engineering techniques. This involves inserting the gene sequences that encode the antibody's variable regions, the parts that bind to the antigen, into expression systems such as bacteria, yeast, or mammalian cells, which then produce the antibody. Single-chain variable fragments, SCFV. SCFVs are synthetic antibody fragments consisting of the variable regions of the heavy VH and light VL chains, connected by a flexible peptide linker. They retain antigen binding specificity and can be used in various applications, including diagnostics and therapeutics. Their modular structure allows for customization of binding specificities, affinities, and functionalities through protein engineering techniques. Due to their compact size and monomeric nature, SCFVs exhibit improved tissue penetration and can be efficiently produced in bacterial or yeast expression systems. These features make SCFVs valuable tools in diagnostics, therapeutics, and research, enabling targeted detection, delivery, and modulation of specific antigens or epitopes. Additionally, SCFVs have demonstrated utility in applications such as imaging, biosensing, and targeted drug delivery, highlighting their versatility and potential for addressing various biomedical challenges. Fab fragments. Fab fragments are synthetic antibody fragments derived from the antigen binding fragment, a Fab region of immunoglobulins, consisting of the variable domains of both the heavy and light antibody chains. These fragments retain the ability to bind antigens with high specificity, making them valuable tools in various biomedical applications. Fab fragments lack the Fc region of antibodies, rendering them monovalent and eliminating effector functions such as antibody-dependent cellular cytotoxicity, ADCC, and complement-dependent cytotoxicity, CDC. Their smaller size compared to full-length antibodies allows for improved tissue penetration and reduced immunogenicity, making them suitable for therapeutic and diagnostic purposes. Fab fragments can be engineered to enhance binding affinity, stability, and specificity, and they have been widely used in research, diagnostics, and the development of antibody-based therapeutics, including targeted drug delivery systems and imaging agents. Their versatility and modifiability make Fab fragments indispensable components in the arsenal of molecular tools for studying and manipulating biological systems. Nanobodies. Nanobodies, also known as single-domain antibodies, SDABs, or VHH antibodies, are synthetic antibody fragments derived from the variable domain of heavy-chain antibodies found in camelids, such as llamas and camels. These unique antibodies consist of a single polypeptide chain containing a single variable domain, making them significantly smaller than conventional antibodies. Despite their size, nanobodies retain high affinity and specificity for their target antigens. Their small size, stability, and solubility enable them to access epitopes that may be inaccessible to larger antibodies, allowing for precise and potent targeting. Nanobodies have emerged as versatile tools in biomedical research, diagnostics, and therapeutics. They offer advantages such as efficient tissue penetration, rapid clearance, and the ability to bind to hidden or cryptic epitopes. Nanobodies have been successfully engineered for various applications, including neutralizing viral infections, targeting cancer cells, imaging specific biomolecules, and delivering therapeutic payloads. Their unique properties and adaptability make nanobodies promising candidates for addressing diverse biomedical challenges. Bispecific antibodies. Bispecific antibodies, BSAbs, are synthetic antibodies engineered to simultaneously bind to two different antigens or epitopes. These specialized molecules bridge two distinct targets, enabling novel therapeutic mechanisms and enhanced efficacy compared to traditional monoclonal antibodies. Bispecific antibodies can be designed in various formats, including IgG-like bispecific antibodies, bispecific T-cell engagers, BiTEs, and dual variable domain immunoglobulins, DVD-IGs. One common application of bispecific antibodies is redirecting immune cells, such as T cells, to tumor cells, thereby facilitating targeted cytotoxicity against cancer cells. Additionally, bispecific antibodies have shown promise in neutralizing multiple pathogens simultaneously and modulating complex biological pathways involved in diseases like autoimmune disorders. Their ability to engage multiple targets simultaneously opens up new avenues for precision medicine and combination therapies, making bispecific antibodies a rapidly evolving area of research and development in the field of biotechnology and medicine. Antibody-drug conjugates, ADCs. Antibody-drug conjugates, ADCs, are a class of synthetic antibodies designed to deliver cytotoxic drugs selectively to target cells expressing specific antigens. These complex molecules typically consist of three main components: a monoclonal antibody that binds to a target antigen on the surface of tumor cells, a linker molecule that attaches the antibody to a cytotoxic drug, and the cytotoxic drug itself. ADCs exploit the specificity of monoclonal antibodies to target cancer cells while minimizing damage to healthy tissues, thereby reducing systemic toxicity associated with traditional chemotherapy. Upon binding to the target antigen, the ADC is internalized into the cancer cell, where the cytotoxic drug is released, leading to cell death. This targeted approach enhances the therapeutic index of the cytotoxic drug, allowing for improved efficacy with reduced side effects. ADCs have demonstrated clinical success in the treatment of various cancers, including breast cancer, lymphoma, and leukemia, and continue to be a focus of research and development in oncology due to their potential to improve patient outcomes. Anticallins. Anticallins are synthetic binding proteins engineered from human lipocolins, a family of small, stable proteins found in the body. These engineered proteins are designed to bind specifically to target molecules, including antigens, small molecules, and protein targets. Anticallins offer several advantages over traditional antibodies, including their small size, high stability, and amenability to engineering for specific binding properties. They can be tailored to bind to a wide range of targets with high affinity and selectivity, making them valuable tools in diagnostics, therapeutics, and research. Anticallins have shown promise in applications such as targeted drug delivery, imaging agents for medical imaging techniques, and modulators of protein-protein interactions. Their versatility, stability, and customizable properties make anticallins attractive candidates for addressing various biomedical challenges and advancing precision medicine approaches. DARPins, Designed Anchor-and-Repeat Proteins. Designed Anchor-and-Repeat Proteins. DARPins are synthetic antibody mimetics engineered from anchor-and-repeat domains, a class of protein-binding domains found in nature. DARPins are designed to specifically bind to target molecules, including proteins, peptides, and small molecules, with high affinity and specificity. They possess several advantageous properties such as small size, high stability, and ease of engineering for various applications. DARPins offer advantages over traditional antibodies, including their modular structure, which allows for precise customization of binding specificity and affinity. Their robustness, solubility, and resistance to aggregation make them attractive candidates for therapeutic interventions, diagnostics, and research tools. DARPins have shown promise in applications such as targeted drug delivery, inhibition of protein-protein interactions, and molecular imaging. Their versatility and adaptability continue to drive innovation in biomedical research and the development of novel therapeutics. Engineered antibody fragments with modified effector functions. Engineered antibody fragments with modified effector functions are synthetic antibodies designed to enhance or alter their interactions with immune cells and other components of the immune system. By manipulating specific regions of the antibody, such as the Fc region, researchers can modulate effector functions such as antibody-dependent cellular cytotoxicity, ADCC, complement-dependent cytotoxicity, CDC, and antibody-dependent cell-mediated phagocytosis, ADCP. These modifications can result in improved therapeutic efficacy, increased potency, and tailored immune responses against target cells or pathogens. Engineered antibody fragments with modified effector functions are utilized in various biomedical applications, including cancer immunotherapy, infectious disease treatment, and autoimmune disorder management. By fine-tuning the immune response, these synthetic antibodies offer new strategies for combating diseases and advancing personalized medicine approaches. Nucleic acid aptamers, chemical antibodies, or synthetic antibodies. Nucleic acid aptamers are short, single-stranded DNA or RNA molecules that fold into unique three-dimensional shapes, allowing them to bind specifically and tightly to target molecules, including proteins, small molecules, and even cells. This ability to recognize and bind to specific molecular targets with high affinity makes them analogous to antibodies, earning them the moniker chemical antibodies or synthetic antibodies. The development and use of aptamers in various fields such as therapeutics, diagnostics, and research highlight their versatility and potential as alternatives to traditional antibodies. One of the key advantages of nucleic acid aptamers is their synthetic nature, which allows for precise engineering and modification to optimize their binding properties. Aptamers can be easily synthesized or chemically modified, enabling fine-tuning of their affinity, stability, and specificity. Moreover, aptamers are typically smaller in size compared to antibodies, which can facilitate better tissue penetration and access to cryptic binding sites. These properties make aptamers attractive candidates for various biomedical applications, including diagnostics, therapeutics, and biosensing. In biomedical research, nucleic acid aptamers have been developed as powerful tools for targeted delivery of therapeutic agents, including drugs, nanoparticles, or siRNAs, to specific cells or tissues. Additionally, aptamers have shown promise in diagnostic assays for the detection of biomarkers associated with diseases such as cancer, infectious diseases, and neurological disorders. Their versatility, stability, and ability to be easily modified make aptamers valuable alternatives to traditional antibodies, offering new opportunities for precision medicine and personalized therapeutic interventions. Non-immunoglobulin, non-Ig, protein scaffolds. Non-immunoglobulin, non-Ig, protein scaffolds are synthetic protein frameworks engineered to mimic the antigen-binding properties of antibodies, but they do not rely on the immunoglobulin domain structure. These scaffolds offer advantages such as smaller size, greater stability, and increased versatility compared to traditional antibodies. They are designed using computational modeling and protein engineering techniques to create binding surfaces tailored for specific targets. Non-Ig protein scaffolds are generated from diverse protein frameworks, including small natural proteins or synthetic peptides, and can be further modified to enhance binding affinity, specificity, and pharmacokinetic properties. One example of non-Ig protein scaffolds is the designed ankyrin repeat protein (DARPin) platform, which utilizes ankyrin repeat domains to create binding proteins with high specificity and affinity for target molecules. Another example is aptamers, which are engineered from the cystatin protein family and offer an alternative to antibodies for various applications. Additionally, adnectins are based on the fibronectin type 30 domain and have been developed as novel targeting agents for therapeutic and imaging purposes. These non-Ig protein scaffolds can be produced through recombinant protein expression systems and can be easily modified to incorporate additional functionalities such as conjugation with drugs or imaging agents. Non-Ig protein scaffolds have found applications in a wide range of fields, including diagnostics, therapeutics, and research. They are used as molecular probes for target validation, imaging agents for medical diagnostics, and therapeutic agents for targeting specific disease pathways. Their small size and stability make them particularly well-suited for applications requiring tissue penetration or targeting of intracellular antigens. Overall, non-Ig protein scaffolds represent a versatile and promising class of molecules with diverse applications in biotechnology and medicine. Among the intriguing developments in this field are aptamer proteins, which are small, robust affinity reagents derived from the cysteine protease inhibitor family of cystatins. They are engineered with high specificity and affinity for target proteins, making them valuable for a range of applications, including research, diagnostics, and therapeutics. Aptamer technology is being developed by Avacta Life Sciences, highlighting the commercial potential and applicability of these synthetic antibodies. The use of synthetic antibodies is expanding rapidly, reflecting their utility across various domains. In research, they serve as powerful tools for protein capture and inhibition. In diagnostics, they are used for detecting infections, cancers, and other conditions. Synthetic antibodies are also emerging as a significant class of therapeutics, driven by their capacity to target specific epitopes, thereby minimizing off-target effects. This specificity is leveraged in designing antibodies to combat pathogens or modulate immune responses, either by activating or suppressing certain pathways. However, despite their potential, the production and administration of monoclonal antibody therapeutics remain challenging, being both time-consuming and expensive. Innovations in synthetic nucleic acid-based delivery are being explored to address these issues, promising to simplify the administration and reduce costs to SARS significantly. An exciting avenue within synthetic antibodies involves the development of biodegradable and sustainable options, such as molecularly imprinted polymers, MIPs. MIPs are designed to mimic natural antibodies' molecular recognition capabilities, enabling the specific binding to target molecules. Recent efforts focus on improving the sustainability of MIP technology, addressing challenges such as the use of non-biodegradable materials and reliance on organic solvents. The shift towards producing nanoparticle formats, MIP-NPs, aligns with a more biomimetic approach, aiming to more closely resemble natural antibodies and thus could be considered true synthetic antibodies from a green perspective. Despite these advancements, there remains a need for further innovation to ensure that the production methods for these synthetic antibodies align with global sustainability efforts. In summary, the field of synthetic antibodies is advancing rapidly, offering promising new tools for diagnostics, research, and therapy. Innovations in this space, such as aptamer proteins and sustainable MIP-NPs, highlight the potential for these synthetic constructs to play a critical role in future biomedical applications. However, challenges related to production efficiency, cost, and environmental impact require ongoing attention and innovation. Stay curious, stay innovative. I'm Luke McLaughlin. If you would like to learn more about this topic, consider reading our full-length articles on biotechnologyreviews.com, or consider our podcast series on YouTube or Spotify at our Biotechnology Reviews channel. Our articles are also available on our LinkedIn newsletter, Biotechnology Reviews.